Now let us discuss about the blood. Blood, we know it's a red colored fluid. It circulates throughout the vascular system in human and other vertebrates. So what is the main function of blood? It carries a nutrient to all part of body. It carries oxygen to all part of the body. And apart from that, it removes waste products including carbon dioxide. So blood, remember, color of blood. What are the properties of blood? Blood, it is red in color. The arterial blood, it is scarlet red due to high oxygen content, while the venous blood, it is purple red because it contains more carbon dioxide. The average volume of blood in human adult, it is about 5 liters. In a newborn, the volume is around 450 ml and it gradually increases with the growth and it reaches 5 liter by puberty. For females, it is slightly less, around 4.5 liters. Blood constitute about 8% of the total body weight. Now viscosity, remember that blood is 5 times more viscous than water. That is primarily due to presence of red blood cells and plasma protein. Blood cells contain fluid that is known as plasma. Now there are three types of cells present in the blood. They are red blood cells, we call them at erythrocytes. White blood cells, they are known as leukocytes, and third are the platelets. What do you mean by hematocrit value? If the blood is collected in a hematocrit tube with suitable anticoagulant and it is centrifuged for 30 minutes at 3000 rpm, the red blood cells settle at the bottom, leaving a clear plasma cell at the top. So the plasma it makes around 55 and red blood cells they consist of around 45% of total blood volume. So the percent volume of red blood cells is called hematocrit value, or it is also known as packed cell volume. Now the plasma, we know it's a straw colored fluid that makes the liquid part of the blood. It contains 91% of water and 8 to 9% of solids. Now what is serum? Serum, it is a clear straw colored fluid that oozes from the blood clot. So when the blood it is shed or collected in container, it clots as fibrinogen is converted into fibrin, trapping the blood cells and forming a clot. So serum is plasma minus fibrinogen. So what is the function of blood? Blood transport nutritive substance like glucose, amino acid, lipid, vitamins absorbed from the GIT tract to different parts of body. It has a respiratory function. Blood transport respiratory gases, oxygen from alveoli to lung to tissue and carbon dioxide from the tissue to the alveoli. It has excretory function. Blood removes the waste products during metabolic activities in tissue and transport them to excretory organs like the kidney, skin and liver for elimination. Apart from that, there is transport of hormones and enzymes. Blood transport hormones secreted by endocrine glands to their target organs and also carries enzymes. The third is the regulation of acid base balance. Plasma protein and hemoglobin act as a buffer, helping maintaining the acid base balance in the body. The last is the regulation of the body temperature due to its high specific heat. Blood plays a role in maintaining the body temperature through thermoregulation. Apart from that, it has a storage function. Blood serves as a source for water and essential substances like proteins, glucose, and potassium, which are required by the tissues, which can be utilized during conditions like starvation, fluid loss, and electrolyte imbalance. Apart from that, blood has defense function that plays in a defense The WBC are responsible for this function. The neutrophils, monocytes engulf bacteria by phagocytosis. Lymphocytes are involved in immunity development, and eosinophils with the detoxification and removal of the foreign objects. The plasma proteins, we know they are the essential component of the blood. Now, the plasma protein can be serum albumin, serum globulin, or fibrinogen. So, the most abundant protein responsible for maintaining the oncotic pressure and transporting various substances is serum albumin. So remember, serum albumin is the most abundant protein, and it is responsible for maintaining the oncotic pressure and transporting various substances. Serum globulin, remember, it is composed of alpha, beta and gamma globulins. So serum globulin, it is composed of alpha, beta and gamma globulins, and gamma globulins, they play a role in immune system by acting as an antibody. The third is fibrinogen. Fibrinogen is a key protein that is involved in blood coagulation. We know that the plasma, it contains albumin, globulin and fibrinogen. Serum is defined as plasma without fibrinogen. So serum is plasma without fibrinogen. So it contains albumin and globulin, because fibrinogen, it is converted to fibrin during the clotting. So what are the normal values of plasma protein? Remember that the serum albumin value is 4.7 gram per deciliter, serum globulin is 2.3 gram per deciliter, and fibrinogen is 0.3 gram per deciliter. The albumin globulin ratio, it is very important parameter. The normal value is 2:1, and it helps in diagnosing liver and kidney diseases. Now, what are the various methods of separating plasma proteins? Remember that the first is salting out method. It differentiates serum globulin into euglobulin and pseudoglobulin using solutions like sodium chloride, magnesium sulfate. Second is electrophoretic method. It separates protein based on the electric charge. The other is ultracentrifugation method. It separates protein by density, also used for determining the molecular weight. Other is gel filtration chromatography. It separates protein by the size using a bed of porous beads. Now the properties of plasma protein, the molecular weight, remember albumin has weight of 69,000 and globulin is 15 lakh 6,000. So the oncotic pressure, plasma oncotic pressure contributes to the oncotic pressure in the blood, with the albumin playing the significant role. Now origin of the plasma protein, they originates in the embryo. They are synthesized by mesenchymal cells. In adult, they are primarily synthesized in the liver with contribution from spleen, bone marrow and other cells. Gamma globulins are synthesized by B lymphocytes. Now coming to the function of the plasma protein, they help in the coagulation of the blood. Fibrinogen is essential for blood clotting. They play important role in defense mechanism. Gamma globulins act as an antibody. They play an important role in transport mechanism. Albumin and globulin transport hormones, enzymes and metals. Oncotic pressure maintenance prevents excessive fluid intake from the capillaries. There is regulation of acid-base balance. Viscosity of the blood contributes to the blood pressure maintenance. It also influences the ESR rate. Now, what is plasmapheresis? An experimental procedure to show the importance of the plasma protein and their synthesis by the liver. It involves the removing blood, separating the red blood cells and re-infusing them with saline. If the proteins are depleted and not replenished, the animal goes into the shock. So they are used for treating autoimmune diseases by removing antibody from the blood. So number of diseases that have been treated by plasmapheresis are the autoimmune disease, bleeding disorders, peripheral neuropathy, neurological autoimmune disorders, neurological disorders affecting the myelin sheath, autoimmune disorder of neuromuscular junction. So they all have been treated by the good posture syndrome. So these are treated by plasmapheresis. Autoimmune disorder of neuromuscular junction. So they all have been treated by the good posture syndrome. So these are treated by plasmapheresis. Our next topic is red blood cells. So red blood cells are other name of red blood cells is erythrocytes. So what are the most numerous elements that are present in blood? They are the RBC. So they are characterized by their red color due to pigment hemoglobin. So their main purpose is to transport respiratory gases, oxygen and carbon dioxide through the body. So what is the RBC count? In an adult male, remember the RBC count is 5 million per cubic mm. In adult male, the RBC count is about 5 million per cubic mm. And in adult female, it is about 4.5 million per cubic mm. So these value can vary based on age, sex and physiological condition. Now the shape of RBC, remember it is biconcave with or a disc shape with thinner center and thicker periphery. So this unique shape provides several advantages. Number one, a large surface area for absorption and removal of substances. Second, rapid diffusion of oxygen and other substance. And third is minimum tension on the membrane when the cell volume changes. The diameter of RBC is 7.2 micrometer. RBC, remember, they are non-nucleated, means they lack nucleus. Due to absence of nucleus, RBC lack DNA and other organelles like the mitochondria and Golgi apparatus. So there is no mitochondria. They rely on the glycolysis for energy production. They don't have insulin receptor. So glucose uptake is not controlled by insulin. The RBC cytoskeleton, it is made up of actin and spectrin that are held by ankyrin to the transmembrane protein. A defect in spectrin leads to condition that is hereditary spherocytosis, where the cell lose their biconcave shape and become spherical. Now what are the properties of red blood cells? When blood is outside the body, RBC tends to form a stack as similar as pile of coins. So this is known as Rolex formation. So this formation is enhanced by plasma protein like globulin and fibrinogen. Now the average lifespan of RBC, remember it is 120 days. As RBC age, their cell membrane become more fragile, especially when they try to squeeze through the small capillaries. So remember, the spleen is known as graveyard of RBC, where the most senile RBC they are destroyed. So that is because the diameter of splenic capillaries is very small, making it harder for older, more fragile RBC to pass through. Now the lifespan is determined by using radioisotopes method. Various radioactive substances like the radioactive iron or chromium, their lifespan is calculated by studying the rate of loss of tagged cells from circulation. From the loss of these tagged cells from circulation, we can know the lifespan of RBC. So once RBC they get destroyed, remember RBC they will release hemoglobin, which is quickly phagocytized by macrophages, particularly in the liver, that is the Kupffer cells, spleen, or bone marrow. Now, degradation of hemoglobin. Hemoglobin is degraded into three components: iron, globin, and porphyrin. The iron it combines with the apoferritin to form ferritin, that is stored and later reused by body. Globin it is broken down into amino acid and enter the protein pool for the later use, whereas the porphyrin it is degraded into bilirubin, which is then excreted by liver through the bile. Approximately 10% of RBC they are destroyed daily in the healthy adults, releasing about 0.6 gram per deciliter of hemoglobin into the plasma. Now coming to the function of red blood cells, they help in transport of oxygen, transport of carbon dioxide. RBC carries oxygen from the lungs to tissues via hemoglobin, which forms oxyhemoglobin when combined with oxygen. RBC also carry carbon dioxide from tissue to the lungs. Hemoglobin binds with carbon dioxide to form carbaminohemoglobin, which account for 30% of carbon dioxide transport. It has a third is buffering action in the blood. It plays a role in buffering action. It plays a buffering action in the blood. So hemoglobin acts as a buffer, helps to regulate hydrogen ion concentration and maintain acid base balance. RBC possess blood group antigens like A, B, which are important for blood group determination and preventing the reaction during blood transfusion. Now, what is polycythemia? And increase in RBC count in response to physiological condition is polycythemia. Now, at birth, RBC count is high, but it decreases after 10 days due to RBC destruction, sometimes causing physiological jaundice in the newborn. Before puberty and after menopause, females have similar RBC count to males. During reproductive period, the count is slightly lower, around 4.5 per cubic millimeter. People who are living at high altitude experience an increase in RBC count due to hypoxia, that is the decrease in the oxygen level.